So good morning or good afternoon, everyone. We're just in the process of filling in, kind of pulling people in from the waiting room. So we'll just wait a second or two before that's complete. I think that's going okay. Okay, introduction. So hi, everyone. My name's John Griffiths. I'm an assistant professor at University of Toronto and a scientist at a research institute in Toronto based in a hospital called CAMH, or the Centre for Addiction and Mental Health. And the institute or the centre at CAMH that I work at is called the Krembil Centre for Neuroinformatics, which is a new outfit, actually. We've been going just a year, but we're really scaling up. And I can tell you, I'll tell you in a second a little bit more about what we do actually, because that contextualizes some of the stuff that you'll hear today. But I wanna just kind of kick off with trying to get some interactivity going in our virtual format here. So, normally if we were doing this as an in-person lecture, then at this point I would ask people for hands up and do a little quiz or just show of hands on basically what people's background are and experience is, so I can, adapt a little bit what I'm talking about and give more info on this or that thing. So I'm going to try and do that here through the chat, if that's okay with people. I can see the chat. Actually, I'm just going to find the button to show me the chat. Bear with me 1 second, because I'm on presenter view. Show me the chat. In fact, I can't see the chat. I really want to be able to see the chat so I can get some feedback from you. Where are we? I know there's a button for this. Talk amongst yourselves for a second, guys. Found it. Okay, good. Okay, so I can see the chat box. So I'm gonna ask attendees. Number one, just say hello. That would be great. Say hello and just a brief hi, I'm Bob, I'm Jane. I'm a biology student, I'm an engineer, I'm a art history major or whatever. So introduce yourselves just so I can kind of get a little mental statistics on who we're talking to here. And as a part of that, this can be part of my little poll, right? So I'd like to know, what's your experience in terms of neuroscience? Because I'll go on in a second to give you the outline for today and the next 3 days. But it'd be useful for me to know if you have some experience in neuroscience, if you're a neuroscientist or whatever, comfortable. If you have zero experience in neuroscience, you know nothing about the brain. And yeah, well, and also neuroimaging, so brain imaging. If you have some experience in that, I'll say as well, you don't need to kind of connect your statements of your experience to your introduction, right? So if you're a little bit, shy about confessing confessing to being a student, which you absolutely shouldn't be, then I guess your name will be in the comment, but you can just say, just write a little thing, experience in this, no experience in this, so on. I'm not really looking at names, so don't worry, that won't get kept on the permanent record. Cool, so I'm getting some feedback here. So hi Chris, biostatistician, faculty of Faculty of Medicine at UT, excellent. Zero neuroscience background, good, thank you for that. Elsa, hi Elsa, evolutionary biology, nice to meet you. Uriel, interest in bioinformatics, no neuroscience, okay, so not so many neuroscientists with us. Paul is atmospheric physics, that's cool. Leah, environmental science, that's cool. Seshu, PhD student biophysics. Emils is, Oh, working with Gunnar Bloom, cool. Yeah, he does interesting stuff. Computer science and psychology. So I'm getting a lot of, I'm getting a mix of actually biologists and maybe kind of physicists, kind of physical science type folks, engineers. Not necessarily all the biologists are neuroscientists. Cool. Sridhar, hi. Amir, hi. Alexander, hi. I'm going to go through the list, but I might stop at some point because it might get long. So apologies to people if you don't get a call out. Hi, Grace. Cool. Hillary, neuroscience. McGill, a computational astrophysicist. Like the sound of that. Chemistry. And finally, hi, Charlotte from psychology. You have experience in neuroimaging. Excellent. Okay. Thanks a lot, guys. That really helps with me. Okay, so let's look to the plan for the next few days. I'm gonna just move some boxes over so that I can see my own slides. Great, so that was me. I'll come back and I'll say one or more things briefly in a second about neuroinformatics and computational neuroscience, which is the kind of work that I do in my group, which leads into the stuff that we'll be talking about in this course. But first up, just the agenda. Okay, so we're running a little late, but that's okay because we have some kind of loose time today. So the plan is I guess I expect most of you guys have been doing some of the other courses from the previous few weeks. And my understanding is that there's been some variation, I know that there's a kind of basic format, but there's been variations on the theme. We're doing some variations on the theme here. So hopefully this won't kind of rock the boat too much. So today is going to be a lecture, just me talking to you with some slides, telling you about some core things that you need to know if you're going to understand and do some interesting computational work in whole brain modeling. Tomorrow, we're going to get hands-on as in using, running some simulations, looking at some data. And this is going to be on SciNet or your other alternative, basically Python environment of choice. We'll talk about tech specs tomorrow primarily, but it's going to be basically a Jupyter Notebook based hands-on session tomorrow. And then the homework assignment is going to be following on from the hands-on session. So it's going to be a kind of modest, fun continuation of the stuff that you'll see at the end of tomorrow. Thursday, there's no session on Thursday, but the homework that you're getting on Wednesday is going to be due. And then Friday, we're going to have a wrap up. I'm going to address some questions, and we're going to discuss some of the better assignment submissions and just generally have a conversation. So one thing to emphasize here is there's a schedule change. And so initially the schedule was Tuesday, Wednesday, Thursday. There was a clash. So it's now Tuesday, Wednesday, Friday. Okay. The Thursday session has been moved to Friday. But the item I have in here on Thursday is just the nominal deadline for you to have submitted your homework. So another few housekeeping points before we get into the content is those of you who are seeking credit for the course, you will need to go to the SciNet course website to register, indicate your attendance. I believe the code for today is 3, and Marcello will be putting a little note in the chat to refresh you on exactly the process that you need to do in order to indicate your attendance. But yeah, as a reminder, you need to have indicated your attendance through the website in order to get credits for the course. And the other thing that you need to do in order to get credit for the course is to submit the homework. Okay, it's not going to be mind-blowingly hard homework, so don't freak out too much about that. And it should be fun. but to submit that is going to be go to the website, and there's a link on the website for homework submissions, which we'll talk about more tomorrow. So again, today is going to be basically neuroscience, and then tomorrow is going to be computational stuff, all of the SciNet details, the homework assignments, etc. That's all coming tomorrow. So don't worry if you find yourself at the end of today like not sure what you need to be doing for the homework, or not sure how to be logging in to run some code. We're not doing that today. We're doing that tomorrow. Great. So I have the chat open, so people can ping with questions, and I'll try to be responsive along the way. You can ping for procedural questions if any of this stuff that I just went over was not so clear to you, and of course, just general questions on the content as we go through. So yeah, feel free to, treat this as we would do a in-person interactive seminar lecture. And if you have questions where something's unclear, or elaborations, or even kind of interesting thoughts, then feel free to chatter away. And I find I've done a few of these online lectures over the last few months, and they're really enriched actually by having a nice, vibrant discussion on the chat. So I will do my best to be responsive to that, but I just want to encourage you to make use of the chat as both a questioning space and a kind of discussion thread. And you can even answer each other's questions if you feel like that's something that would be helpful. Great. So let us go back to the lecture. And so another procedural point, this is going to be more relevant to tomorrow, is there is a GitHub repo for the course. This is I've actually been moving some stuff around recently, so this doesn't have all of the materials in yet, but it will do tomorrow. So, if you're super keen, then you can check this out, not right now, but in probably a few hours, and you'll find more stuff there. But really, it's tomorrow when you're going to be accessing this GitHub repo and looking at the practical materials for the session. Good. So, I'm gonna go over 4 things over the next roughly an hour. We'll do an hour to an hour and a half. So we'll do probably the first 3 of these, and then take a break. I'll take a break at roughly halfway through, depending on exactly what time that is, and then we'll continue. And the break will be a good time. Well, it'll be a good time for you to stretch your legs and grab a cup of tea, but we'll also, at that juncture, we can have some back and forth on questions if there's anything that I haven't been addressing along the way. So I'm going to talk to you. I'm going to give some general comments on neuroscience and computational neuroscience, and where the type of modeling that I'm going to be telling you about fits in the broad scheme of things in computational neuroscience, because there is a pretty broad scheme, and we're going to be looking at one specific corner of it, and then I'll go over the ins and outs in an introductory fashion of whole brain modeling, and then I'll wrap up with some applications, which is always interesting. It's kind of the kind of things that people do with these techniques, and specifically in the clinical domain. Now, if you if you were to leave right now, or another way of saying is that if I was to give you the 30-second summary, I decided maybe be a bit more than 30 seconds, but the rapid summary of what whole brain modeling is, and the message that I want you to take away from this session, I'm going to summarize it right now. So what we're doing, so first let's start with this phrase whole brain. Okay. In neuroscience, and in computational neuroscience specifically, one of the way in which people try to model and describe neural organization and neural activity mathematically and computationally basically falls into roughly three types. And the type is, broadly speaking, the spatial scale at which you're trying to describe things. I'll go over those spatial scales in the next slide in a little bit more detail, but the take-home message to start with is where we're coming in is in the middle, basically, in terms of spatial scales. So the level that we use when we're thinking about modeling large-scale brain activity, large scale as in at the scale that you would maybe see structures and patterns in human brain imaging. And really, the whole brain is a good description of it. We're talking about activity patterns that span at least most of the spatial extent of the brain, front to back, or frontal lobe to occipital lobe, subcortical structures, etc. When we're talking about modeling stuff at that level, we aim for what we call a reduced or a mesoscopic scale description of the individual units. The reason for that being, we just don't have the computational power and the really the right kind of questions and the right way of understanding simulations, full stop, to be modeling the whole brain at the level of individual cells, which is what I'm talking about when I say physiologically detailed microcircuit models here. So we're looking for a level of abstraction that takes us away from describing every cell or every neuron in the brain, and even every subcompartment of every cell or neuron in the brain, which is what people who work on cell level models do. We're looking for an abstraction that gives us something more like, from a physics point of view, we think of as a mean field description, or a reduced description that says for this set of cells, which is going to be hundreds of thousands of cells that maybe you can find in a patch of tissue around about a cubic millimeter, right? So a patch of brain tissue that's a cubic millimeter is going to have a lot of stuff in it, in terms of cells, like many hundreds of thousands. So rather than enumerating each of those individual cells and all of the parts of those cells, we're going to say, okay, we're going to treat this patch of tissue as basically an individual unit in itself, a mesoscopic scale individual unit, and look for a formalism and a way of simulating the activity that's just at the level of the average activity of that patch, rather than the many thousands of cells that you can find inside it. So we find our reduced description at the unit level, and then the second part is to construct network models or a network level description where our individual units, our mesoscale reduced neural population individual units, form the basic units, or specifically the nodes in the network of this large scale network that describes the connectivity structure of the brain at the mesoscopic spatial scale. So this, and I'll be drilling down on each of these in the next half an hour or so. So don't worry if it's not super clear at this point, but just to explain this figure, we've got, this is a three-dimensional representation of a brain network where each of the red dots are essentially brain regions at a certain, essentially coarse-grained spatial scale of definition of a brain region. Like we could have more red dots here if we were to have a finer grained spatial scale, and often we do. But at this level, which is roughly about 100 nodes here, or 100 red dots, each red dot represents a brain region, and each of the green lines is the anatomical connectivity, basically the wires that interconnect those brain regions. So that's our representation in 3D, as a three-dimensional embedded graph. We also just represent that as a connectivity matrix. That's just telling you what the actual numbers are in the connection strengths between the red dots here. And then this little thing on the bottom is just an example of the type of simulation that that's the endpoint of putting these two things together, the mesoscale reduced description to give us the units, and the network structure to give us the network, is we basically turn the numerical crank, solve our differential equations. I'll be talking more about that in the next few slides, and generate simulated activity patterns. And the activity patterns that we're simulating here are roughly on the kind of, on the spatial scale that we typically are aiming for and measuring with human brain imaging. So it's very commensurate with the type of information that we get from human brain imaging data. And one way of just thinking about this is we're just simulating your imaging data, full stop. And that's, that is what we're doing with whole brain modeling. Basically, we're trying to simulate brain imaging data. We're not trying to simulate cellular physiology data, things that you measure at a very small scale, but stuff that you measure at a relatively large scale. And then the final part of this picture is obviously validation, right? So these are models that they represent theories. They are only as strong as the theories and the models that we put in those boxes, essentially. So obviously they're representations of reality, so they need to be validated, and they need to be useful, as the kind of phrase goes. So the way that we do that is we make empirical measurements. As I was talking a lot about neuroimaging. When I say neuroimaging, I mean that in a somewhat broad sense of primarily human functional magnetic resonance imaging, human EEG, and human MEG. So human EEG, or electroencephalography, is what this figure on the bottom right here is showing. You're probably familiar with this. You've probably seen this before, but you can measure extracranial or electrical or magnetic fields that are generated by the brain with sensors placed on the scalp. And we can do that in non-human animals, as well as primarily humans. Generate time series, generate things like power spectra, and then we want our models to be accurately reproducing the patterns that we're seeing in our empirical data. And one of the ways that one of the things that we want to come out of this process of comparing models against empirical data is getting an idea of what the correct parameters are in those models. So in other words, estimating the physiological parameters that we don't know from measurements, but we express within our models, and we try to learn them by fitting the physiological models to empirical data. So that's whole brain modeling in a nutshell. And as I said, if you could tune out, and if you remember this slide, then you got the gist for today's session, but not the richness. Okay, I have a question before I go on. This is just a procedural question. So I'm sharing my slides. I believe I'm sharing my slides, but not my screen. My screen has some Zoom things on it, as in I can see a Zoom chat and a Zoom images, my face basically. I just want to know, yeah, you can only see my slides. Good, that was what I was checking, because that would be kind of ugly for you to be seeing my chat box. Good, so that's the summary, the rapid summary of whole brain modeling. Now I want to kind of situate this a little bit more in the field of computational neuroscience, which is what, is the field that we're working in here. So this is a sketch that I have that I find quite useful to give the lay of the land for computational neuroscience. It's a very broad church. So computational neuroscience is, in a way, it reflects every subfield of neuroscience. Like every subfield of neuroscience where there's data generated, there's some corresponding complementary piece of computational neuroscience that's trying to understand those data. But it's also in a way broader than that, because it extends into domains that are not necessarily traditionally core neuroscientific domains, like mathematics, computer science, machine learning to some extent, and various technical domains, basically, where we need to make use of theory and we need to make use of computational tools. So computational neuroscience is a very broad field. Broadly speaking, we can group the type of things that people studying computational neuroscience into two rough areas, which I'm representing here on the left and right hand side. And it could summarize basically as, on the left, I have a couple of not exactly synonymous, but related terms like cognition. Another way of describing is like information processing, coding, representation, computation, understanding, okay, high level cognitive processes. These are one flavor of things that people are interested in computational neuroscience. On the other side of the divide here, I've got a group of research areas that I'm broadly describing as dynamics. And the dynamics here are, they range in spatial scales, and they range in their level of abstractness, in abstractness relative to the biological, relative to biological concepts, let's say. So at the smaller spatial scale and perhaps like the less abstract level, we have things like tissue and membrane biophysics. So ion channel fluxes across membranes, how neurons fire, how they generate resting potentials, and all of that really boils down to electrical engineering of cell membranes. Things like control and nonlinear dynamics, how do we understand systems that display nonlinearities and systems that are complex networks, and how can we understand that at if you like emergent spatial scales. So I have a kind of axis here on the right hand side. It's broadly going from left to right, or from the middle to the outer part, is going from increasing in spatial scale. And right on the right hand side here is what we would describe as the mesoscale. I'll describe this a little bit more on another slide in a minute. And this is to really kind of summarize where we are on this picture. We today, thinking about whole brain modeling and large scale brain networks, are thinking about things at the level of the mesoscopic spatial scale, but we're thinking about things in terms of dynamics rather than information coding or representation. So we're really trying to understand really the kind of the mechanics of activity generation and propagation and functional organization at mesoscopic spatial scales, and using some techniques, or in a way like the mathematical foundation it has a flavor of statistical mechanics to it. Statistical mechanics in the sense of, we could say, trying to understand trying to understand a gas in terms of emergent properties like pressure or heat, rather than microscale properties like the movements of individual particles, individual atoms. That's a useful analogy for what the approach that we're taking with mesoscale modeling. And just final point on this sketch is that I have these two domains in computational neuroscience, thinking about information coding, thinking about dynamics, and really the holy grail in computational neuroscience is to bridge this explanatory gap, basically, bridge these two domains and try to understand things like information processing and representation in terms of mainly more concrete things that we can define and we can see at the level of dynamics and at the level of neurophysics. So that's definitely the holy grail. We won't really be trying to tackle that today, but it's a useful thing to know if you're interested in the field. And today we'll be here on the right-hand side. Great. So I talked about spatial scales here. I'm going to give you a little bit more on spatial scales to hammer the point home. So this figure is going from the left, bottom left to the top right. These are increasing spatial scales. I realize, and I've given this talk a few times, and I keep saying this and not changing it, but the bottom right here should be microns, not millimeters. So I apologize for that. But so what we're doing is we're going from the micron level or the single cell level up through various spatial scales. And these are spatial scales that are often broadly defining different different levels of examination in terms of spatial scales in computational models. So at the level of microns and at the level of individual cells, this is the micro level. And these, what we typically describe this is, well, the micro level, micro scale, and sometimes we use the phrase microcircuits, which would be, if we just go up to the second ones, neural populations, these interesting looking hairy things are actually visualizations of densely packed cell bodies in a cortical column. And the different colors here representing different layers within a cortical column. We can discuss a little bit more on cortical columns if people like. I guess I realize I'm assuming a little bit of knowledge there, but I'll leave that for now. Feel free to kind of ping some questions at me about, John, can you tell me, you seem to be assuming that I know what a cortical column is, etc. We can kind of iterate on that as we go ahead. But just to wrap up with the spatial scales here, so our two on the left-hand side here, this is typically what we call the micro scale. Now going up to around around this area, say between millimeters to centimeters, this is what's typically described in neuroscience as the mesoscopic spatial scale. And then going up again on the order of centimeters to tens of centimeters is typically described as a macroscopic spatial scale. So we have micro, meso, macro. You'll see me using the term meso quite often here and there in these slides and in things I describe. One of the key things to emphasize, though, is that these borders between these nominal spatial scale definitions are somewhat fuzzy. They're not concretely defined. So particularly like the transition from micro to meso and the transition from meso to macro, basically, if you just look what people say and people publish and what you see in the literature, you'll see a kind of a distribution of definitions, essentially, of like where meso is defined relative to micro and where macro is defined relative to meso. And to some extent, that reflects things like conventions within different subdisciplines and so on. Great. So we have these different spatial scales, and I mentioned that with whole brain modeling, one of the things that we're aiming to do is really simulate brain imaging data, human brain imaging data. So these levels that I've got here, the level of regions, the level of large scale networks. Actually, these three pictures I have in here, you can probably tell that on the right hand side we have a whole brain or a whole hemisphere, whole cortical hemisphere, and then I'm actually just zooming in on the same image for these other three on the left of there, and this image here is what we call a surface representation of a brain. So it's actually a mesh. You can see from the zoom in that we have these little kind of mesh vertices and edges are represented, and your level of what we call local patches would be something like a few, a couple of square millimeters that you could paint on a cortical surface. And then we zoom out, then this would be something more like what we call a brain region or a brain parcel, or something like a Brodmann region type size. Again, you can see on our cortical surface. And then zooming out, we're thinking more about the holistic whole brain network structure. So we're going to be in this middle to right hand side of this image today, but I want to give a nod to what people work on in other domains at the smaller spatial scales, and to some extent that does feed into and inform, constrain, define the way we think about things at the meso and the macro spatial scale. So I mentioned earlier that this is the typical techniques that we're thinking about here are human brain imaging techniques. This is just some images, what those look like if you maybe haven't seen them before. This is an MRI scanner, or that angelic looking MRI scanner. So when we talk about fMRI or structural MRI, then these are the images that you can obtain non-invasively from healthy subjects and from patients who are sticking their head in this giant donut here. And then I also mentioned EEG and MEG. So EEG, we had a figure back there as well. So EEG, again, I think people are probably often familiar with this, but really it's basically like a swim cap that has a bunch of holes in it that where you place these sensors, these little flat discs that are the EEG channels or the EEG electrodes that measure electrical activity. And then this guy in the middle is called a magnetoencephalography device, which is the magnetic equivalent of your electroencephalography device here. And the way that MEG channel systems work, or at least conventional MEG systems, because newer MEG systems have some slightly different physics, but conventional MEG systems, this big hairdryer type thing, you can see the little hole here, and you can tell from the chair, like you sit in the chair and you stick your head in the hole there. And this big hairdryer thing, which we would call this a dewar, is filled with liquid helium. And the liquid helium super cools some superconducting quantum interference device sensors that allow very sensitive measurements of magnetic fields. So MEG is a really powerful technique, and as you can imagine from how I just described, quite a expensive research technology, but it allows us to get some very interesting and important measurements of meso to macro scale human brain activity at relatively fast temporal scales. So EEG and MEG, you can measure things. Actually, this figure on the right-hand side shows this. The x-axis is temporal scales in terms of time. The y-axis is spatial scales, both on logged axes. And you can see basically being further over to the left is better in terms of temporal resolution. Better as in you can see activity, that you can see faster activity. So EEG and MEG in this box here relative to fMRI is doing pretty well in terms of its temporal resolution because it's further over to the left. So we can see activity with EEG and MEG at the temporal level of milliseconds or even a sub-millisecond temporal scale. We don't typically need to go under a sub-millisecond temporal scale. Neural activity more or less evolves on the temporal scale of milliseconds. So we don't generally need to see things at, say, thousands of kilohertz resolution. 1 kilohertz or sampling at 1000 hertz is good. 500 hertz is, and 250 hertz is often acceptable. And these are easily achievable with EEG and MEG. fMRI, on the other hand, is slower, kind of notoriously somewhat slower because it's measuring blood flow rather than a direct measurement of neural activity. But as you can also see in this figure, it does a better job in terms of spatial scale, as in it extends further down this little fMRI box here. So it's not so far over to the left. So we're seeing things at the temporal scale of seconds with functional MRI. Blood flow fluctuations that respond to neural activity changes. That's what we're actually measuring with fMRI, or blood flow and blood oxygenation. But we can see with a finer spatial resolution, so we can see down to the millimeter spatial scale broadly with fMRI. Sometimes we can go a bit below, often we're a little bit above that, but that's the level of resolution that we can aim to achieve. Great. So that was it for spatial scales. I want to repeat and encourage people to throw in questions if anything's unclear, or if you have some interesting thoughts that you would like me to expand on. I have the chat box in front of me, so I can see your questions. Okay, so moving on, neural population models. So I described this a little bit earlier. I described some of the rationale and what we're, the way we think about this. And I mentioned a little bit this idea of emergent behavior, but I'm going to repeat this and reinforce it with this little video. So the idea here is that, as I said before, we're not, with mesoscale whole brain modeling, we're not trying to describe the activity of individual cells, but we're trying to find a level of description that describes collective activity of large numbers of cells, thousands of cells, roughly at the spatial scale of millimeters, mesoscopic spatial scale. So this idea of collective activity, we kind of need to justify it to some extent. And there are a few ways of justifying it. One way of conceptually thinking about it is with these videos that I'm showing you here. So the analogy is that the individual neurons in our cortical column, for example, on the right-hand side, would be our individual birds in the flock, or insects in the swarm, that type of thing. And the collective variable, the mean field, the neural population, right? That's in some sense the you can think of maybe as the center of mass of these bird flocks. So you can think of and you can define, and it's reasonable to understand the entity that is the flock of birds as a kind of emergent unit that has some center of mass, let's say, some location in space, and it moves around. I mean, sure, its shape changes and its variance changes, its spatial structure changes, but to the extent that we can think about that as an individual, if you like, emergent entity, emergent quantity, that's the kind of thing that we're looking to do with neural populations as well, right? And another useful analogy that I think of for this is trying to see the forest for the trees, right? The expression is like, not being able to see the forest for the trees if you focus too much on the details. So we're zooming out a little bit in terms of the details, as in, quantifying and representing the activity of individual cells and individual units at the microscale. And we're zooming out so that we can see the macro scale behavior and look at larger scale emergent patterns. So at the level of the individual units, I'm going to talk about mean fields. I'm going to talk about neural populations. You can use this bird flocking analogy. It isn't perfect. We can talk about the failures of that analogy, but it takes you a long way to thinking about what emergent behavior or population level behavior, what we're aiming for here is. Now, getting down to the details. So I'm talking about neural populations, and there are a number of different models of neural populations, a number of different ways that neural populations in the field, in the literature, are described mathematically. I'm listing a few of them here. There are many missing. There isn't really time here to drill down on the relationship between these and why why there's so many, and there's many more that aren't listed. But just to flag that there are a number of different approaches to describing neural population activity, and one of the ways that's one of the categorizations that we use is phenomenological versus biophysical. And broadly speaking, phenomenological are less grounded in the physiological structure of neural brain organization. So they're more aimed at describing the the phenomenological, or the kind of the patterns that you see without being overly constrained on defining things in terms of things like wiring principles that we know to be the case. So yeah, biophysical and phenomenological neural population models. I'm going to focus on biophysical ones here. And I'm just going to talk you through one of the kind of principal architectural features of neural population models, and one of the types of neural population model that we'll look at tomorrow. So neural population, also this is kind of somewhat synonymous, neural mass models, is one of the key features of neural mass models. Is what we're trying to do is we're trying to describe the activity of a neural population, and we break that down into two principal operators, or two principal operations, and it's basically an input-output operation. One of the funky terms that's used to describe these two is a wave-to-pulse operation, and a pulse to wave operation, and a wave to pulse operation. Now, the reason for these terms, wave, pulse, and wave. So, pulse here is talking about a firing rate. So it's saying at the population level, we have some neurons that are providing an input, and the input is coming in as spikes. So kind of binary on-off signals, if you like. And what happens is that's translated into, so this firing rate input is translated into a continuous valued variable that is the population membrane potential. So it's like the voltage level of the membrane. So that's what the pulse to wave is really. The pulse is the firing rate, and the wave is the membrane potential. And the kind of mathematical form for the pulse to wave operation is a alpha kernel, or this kind of gamma function, skewed Gaussian type function over time. But what this is giving you is the impulse response of the synapse, or the synapse is at the population level, to a unit impulse. So we have a rapid early response, and then a slowly exponentially decaying response on the order of a few seconds. Our second operation here is the wave to pulse operator, and this is essentially describing the output. So this is saying, for the population membrane potential, how does that get translated into a output firing pattern? Because the way neurons communicate with each other is they spike, and when we're trying to describe them at this mesospatial scale, we don't think about individual spikes, but we think about firing rates, and particularly we're thinking about things at firing rates at the population level, rather than the individual cell level. So the functional form for kind of mapping the population membrane potential to the population firing rate is this sigmoid function that we call the wave to pulse operator. And this just basically instantaneously converts whatever the value of the membrane potential is to a firing rate. Now I can go into, and seeing as we don't have so much time for this, what I kind of go into in the next slide, but I'll not go into the details here, is the kind of derivation of this sigmoid and why we use this sigmoid function. It's to do with it's basically sum of smooth sum of heavy side functions that are varying a little bit in their inflection point, bottom line. But I won't go into the details there. Final general point though on this type of representation is that the bottom line is with the pulse to wave operation, or this synaptic kernel, is we can write it in terms of an integral, but we generally think about it in terms of an equivalent differential equation representation. And we don't need to, I don't need you to understand these differential equations right now, and I won't go into them. I won't kind of unpack them too much. We will encounter them again tomorrow. But the key message here is this is what we're using to simulate our neural population activity. We're using a set of differential equations that's describing the activity of our neural population. That again is a reduced or a mean field representation of the activity of the collective activity of large numbers of cells. Great. So another general point on the structure of these neural mass models is what I showed you just now is the typical architecture of an individual neural population. Now, generally with these type of models, what we actually do is we have several neural populations, not just one, and each of those several neural populations collectively describe the organization of a local patch of tissue, such as a cortical column, and particularly the kind of wiring structure of that patch of tissue. So the most common types of models, almost all of the models that are relevant and that we're going to be thinking about here, they basically summarize your neural population, your kind of cubic millimeter or square centimeter of cortical tissue, in terms of excitatory neural populations and inhibitory neural populations. And that's the level of simplification that does a good job of characterizing the principal types of dynamics that these neural populations exhibit, that we can measure at these meso and macrospatial scales. So we think about populations of excitatory and inhibitory neurons and how they're connected to each other, their local wiring diagram. So this figure here is from an excellent review from a few years ago. It's just showing some of the variation in the types of local wiring diagrams that have been used and studied and proposed to model this kind of neural population activity. Like two I'll point to you. One is the Wilson-Cowan equations, which are to some extent the canonical type of mean field neural mass model. So what this little diagram here is showing you is saying there's a neural population that represents the excitatory cells in our cortical column, and there is a neural population that represents the inhibitory cells in our cortical column. And the excitatory cells, as you can guess by the name, they excite other neurons and other neural populations, and the inhibitory cells inhibit other neural populations. So the wiring structure that here is basically we have the excitatory population exciting the inhibitory population and also exciting itself with this self-feedback, and the inhibitory population inhibiting the excitatory population and also inhibiting itself with self-feedback. So that's it. That's basically all you need to understand about what the type of architectures we're talking about here. They get a little bit more complicated, but only a little bit. So the second one I just want to point to here is this one, which is the Janssen-Ritt model, which we will be looking at tomorrow in more detail. And the idea here is to use more than one excitatory population. And the rationale for this, if people who are neurophysiologists, and there aren't so many of them here, but they are quite aware that there's more than one type of excitatory cell in the brain and more than one type of excitatory cell within a cortical column. And so having two excitatory populations here, what we're actually doing is we're representing some of the excitatory cells that you get in a cortical column in one of our populations, and some of them that you get in a slightly different part of the cortical column that are slightly different cell types and have slightly different projection shapes with our second excitatory population. And specifically in this topology here, I'll actually show you this with the next slide, the Janssen-Ritt model. So here we have our two excitatory populations are the pyramidal cells and the spiny stellate cells, and our one inhibitory population, we're just calling inhibitory interneuron here. And in terms of the wiring here, so the pyramidal cell population is in the middle. So the pyramidal cell population is having is feeding forward to and receiving feedback from the spiny stellate excitatory population, and it's also feeding forward to and getting feedback from the inhibitory interneuron population. But those two aren't talking to each other, the second excitatory and the inhibitory population. They're only talking to the pyramidal cell population. So that's a kind of canonical schematic that is commonly used to summarize the average wiring structure within a cortical column. And the equations on the right-hand side here are the ones I was just showing you with the sets of second-order differential equations describing each of these three populations. Great. And then the final thing with this type of model that I want to emphasize is oscillations and the role of inhibition in generating oscillations. So in general, I mean, in brain systems, but in all kinds of, physical systems, engineered systems, just general, just mathematical like fact that you need negative feedback to generate oscillations. So that's one of the key contributions of the inhibitory population in these schematics is it's feeding back or kind of, pulling, giving some pull. But you think of it a bit like a spring in a way. It's serving a little bit like the kind of friction component of a spring, and it's pushing back on our excitatory population and generating some oscillatory activity. And one of the ways that we represent this is in terms of a power spectrum. So these are just three examples of a power spectrum. We'll look at these tomorrow, so it's worth being familiarized with them now. Basically, it's a profile of the frequency structure that some time series is showing. So the x-axis, I'll just take the one in the middle here. The x-axis is frequencies. By the way, I haven't checked if you guys can see my arrow. I'm hoping you can see my arrow. If not, some things may have been less clear than they otherwise could have been. But in any case, we're on the Robinson model figure here. And on the x-axis, we have frequency in logged units. So we see 1 hertz, 10 hertz, 100 hertz, and then power on the y-axis. So the line is showing you the profile of basically the relative amplitude or power, but the relative amount of energy in the signal at different frequency scales. And we can see here, this is something that's very typical and that we see a lot in human EEG activities. We have a peak in the power spectrum at 10 hertz, or what we call the alpha rhythm. So these power spectra, these frequency plots are pretty important for a lot of things, and we'll be seeing more of them tomorrow. Great, so that's it for this section. And I saw a few little messages that there's some discussion in the chat. So I'm just going to pop over to the chat box and see if there's anything I need to ask. Can you have resonances? Beautiful. Yes. So resonance is exactly the type of thing that I'm talking about when I'm talking about these generation of oscillatory feedback. And in fact, these models here, I didn't really explain it, but one of the key ways that we study these, really powerful ways that we can study these, is we linearize the system around a stable fixed point, and then we obtain a linear expression for the power spectrum, or a transfer function for the power spectrum. And the poles, if we represented it in a polar representation, but the peaks in the power spectrum representation, these are the resonances in the model. So this system is basically being driven by uniform white noise and is generating natural frequencies, oscillatory frequencies at these peaks, and these are the resonant behaviors. And if we stimulated at or around these frequencies, then we would see things like Arnold tongues and preferential responses to frequencies around the resonant frequencies of the system. Cool. So magnifications of energy. Yeah, magnifications of energy, that will be the kind of thing that I'm talking about with stimulation. Do we have any other questions? I don't think so. Good. Okay, so we'll move on. So I talked about the units, talked about neural population models. Those are, and if we think about the schematic that I showed you at the start, these neural population models give us the elements or the nodes in our network. So now let's talk a little bit more about the networks. This image here, I've shown you a few of the, I showed you a few pictures like this that are like a cortical column. Of what that actually means, or to help you out with that, is that this picture on the left hand side, this is from an MRI image. This is what we call coronal section of a brain, and the little box here is showing you essentially what you would see if you zoomed in and looked at this bit of brain, if that was possible. It's possible with some new visualization tools like the Jülich brain or Big Brain, but it's generally not easy to do. You certainly can't see these things on a normal MRI, but this little box here, this is where these neural population, these cortical columns that I've been talking about kind of sit. They sit on the cortical surface, and the cortical surface is actually a thin crust. It's really like you can think of the cortex as, like I say, a beach ball that's got a little bit of thickness to it. The surface, or the kind of the boundary of the beach ball, and there's been scrumpled up, and those scrumples are what we call gyri and sulci. So this is where a neural population is, and if we think about now the connectivity structure, because we'll talk about networks, we're interested in the network representation of this. Each of these points in the brain are the bit on the inside of this white line here. This is the white matter. So this part of the brain is actually all wires. It's all axons and axonal fiber bundles that interconnect different parts of the gray matter. And I'm showing it with a different contrast. This is from diffusion MRI tractography, just trying to show you what you can see here is we have this network of fibers that they kind of send projections out to the outer parts of these cortical gyri. They're kind of like, they're a bit like a cauliflower. They're a bit like the outer part of a cauliflower. So all of this stuff in the middle is network, is wires. So, and we can image that with non-invasive brain imaging with tools like diffusion MRI tractography. So we can reconstruct these fiber pathway trajectories, get some nice images like we see here. And this has led to subfield in neuroscience that's been termed connectomics, is by analogy with genomics. So the idea is the connectome is the map of connections in the brain, the kind of complete map of the brain's connection wiring, in the same sense of the genome is the complete map of the base pairs in human DNA. So we can go into, we can have another discussion, now isn't exactly the time, on exactly what it means to have a complete map of the connections of the human brain. But that's the idea, and we use the phrase. It's used at the micro scale in terms of individual cells, and it's used at the macro scale in terms of what I was describing more with you, with using non-invasive tractography to reconstruct these fiber trajectories within the white matter, and then using these brain parcellations, or these kind of categorical definitions of brain regions, to give us the connectivity structure at that macroscopic spatial scale. Just realized my computer isn't plugged in and I'm getting a battery warning, so I just need to do that. Okay. Right, so this is what we think of as macroconnectomics, looking at the connectivity structure of the brain at the macroscopic spatial scale. This is used for, there's a real industry in brain imaging looking at the graph network properties of large-scale brain networks. There's been big, heavily funded projects like the Human Connectome Project, trying to improve the tools and the measurement techniques that we use in brain imaging to study connectomes at the macro scale. And coming back to our modeling, and the focus of this course is, so the type of models that we use when we're trying to describe whole brain activity, doing whole brain modeling, one of the terms for this is connectome-based neural mass models. So it's putting our neural masses, our neural populations, into a connectome. We can do fields or continuous and continuous domain neural representations of neural populations as opposed to point processes, which is very interesting, but it's rather an advanced technical point that I won't go into too much more here. And one of the principal tools that's emerged over the last few years for doing this type of modeling is a software platform called the Virtual Brain, which I have some involvement in the development and the kind of usage of. And it's the folks who came up with this are my former boss, Randy McIntosh, who's a scientist at Baycrest, and his colleagues Victor Yersa in Marseille, and Petra Ritter in Berlin, and a large extended team of people who've really contributed to building this tool. But the idea with the Virtual Brain, or TVB, is it really pulls together these different components that I've been describing that are the ingredients of a connectome-based or large-scale brain network model. Is we have our just working from the left and kind of going around in this circle here, we have the cortical surface defining our brain regions. We have the connectivity, or the connectome, which I just described to you, giving us the wiring structure. We have our neural population models that describe the dynamics of neural population activity at the mesoscopic spatial scale. And we put those together to give us our brain network. And then we turn the numerical crank, or, simulate the activity according to the differential equations that we've specified. This generates synthetic or simulated signals, which typically we are interested in simulating signals from neuroimaging data like EEG, MEG, fMRI, and then we want to compare these against experimental data. So TVB is a very nice tool. We're going to be using it tomorrow, which is why I'm elaborating on it right now. And it allows us to easily put together these different components of our brain network model, the network structure and the neural population model particularly, and easily generate simulated neural activity. So we'll be seeing more of TVB tomorrow. It will be our friend. And this is the type of thing that we can do so we talked a little bit about oscillations. I showed you this movie earlier on actually, but just to give you, to circle back on it. So now I've given you the more of the context. So this movie up on the right-hand side is showing you oscillatory activity in a network of neural masses, which is represented on the bottom right here. And each element in that network is described by a neural population model that has a wiring structure that's indicated by the schematic in the middle. And this is generating oscillatory activity, which we can see in the time series on the bottom right-hand side, that has a power spectrum. We can see in the power spectrum right on the bottom right of around 10 hertz. This is a connectome-based whole brain model of cortical alpha activity. Great. So doing well. We have 10 minutes more to go, and I'm going to conclude with some of the clinical applications of these tools. But I want to circle back again and just see if anyone has any questions about any of the material so far. There was a great question from some, okay. I didn't quite catch who was asking the question about resonances, but that was a great question. This is really cool. Glad to hear. So people can throw in some questions now, or they can throw in some questions and I'll take another look at the end. Functional brain modeling. Yeah, so functional brain modeling is what we're going to be looking at. We're going to be doing simulating neural activity that we can use to create correlation networks that are typically called functional connectivity. So that's generally, those are often called functional brain networks, and are related to functional MRI. So maybe that's what the question there is asking about functional brain modeling. If that's not, then if you can elaborate and I can help you out. But yeah, definitely one version of functional brain modeling is the type of thing we're going to be looking at. Okay, so I will crack on with the survey of clinical applications. Okay, I have another quick question before we do that. Do you use a network itself as in the topology? Yes. I mean, I guess when you say the topology, you don't mean topological properties per se. So we're not using kind of topological graph metrics, but we are using a network. We're using networks that are roughly 100 nodes representing all of the brain, mostly cortex, but some subcortical regions. Those 100 nodes, those are what we call a brain parcellation in neuroimaging. And it's similar to, if you've ever heard of Brodmann areas, it's like a 21st century kind of Brodmann area. How big are they? When you're talking about a parcellation of 100 nodes, you're looking at each node is representing a piece of tissue that is a couple of cubic centimeters, maybe 3 to 8 cubic centimeters, varies for different nodes. We can use different parcellations that are larger. So we could, for example, we use a 1,000 node parcellation, where each of the individual nodes is then maybe down the level of 1 to 3 cubic centimeters rather than 5 cubic centimeters. So this is one of the things about this area of neuroimaging. It's a kind of a frontier of the scientific field to some extent, is what's the right level of spatial scale to be defining your parcellations at. And there isn't really a right answer to that. The only answer is really we can use parcellations at smaller or larger spatial scales that give us smaller or larger networks. And yeah, so the question is, can we go deeper? Yeah, so we can crank it up to thousands of nodes. We don't typically go larger than thousands of nodes. People definitely do this. I mean, it's numerically possible. It's harder to do with delays, but it's certainly within the realms of a modest simulation on a modest RAM capacity of a decent laptop, and certainly a SciNet node to do a couple of seconds of simulation of a network with, say, 20,000 brain regions or 20,000 nodes. But typically we're not up at 20,000. Typically we're more in the range of 100 to 1,000. And we can discuss more the reason for that. There are good reasons, but there are also just limitations. It is harder to do simulations with larger networks, but it's very much like a fluid area. You can choose where to go. There isn't really like a right or a wrong scale. There's just scales that people tend to use because they're easier or because they correspond naturally to some of the types of empirical data that we might be interested in one particular context. Very good questions. Okay, so I will crack on with my survey, and then I will wrap up for today's session. So I'm going to talk about three areas where this type of modeling has been used in the clinical domain. I'll actually focus on one of them and just skip through the other ones fairly quickly. But here we go. So one of them is auditory attention. And there is people, if there's anyone here, I know there was someone in psychology who might be familiar with this. There is a phenomenon. So with EEG, one of the things that we look at are called evoked responses or event-related potentials, which is shown by these little squiggles on the brain in the middle here, but also on the right-hand side. And they're essentially like a time series of brain activation that are differential for different types of stimuli and different types of cognitive phenomena. And auditory attention is a nice example where we can get a distinct evoked response or a distinct difference in the brain response to simple auditory stimuli, tones that are either expected because you're hearing lots of them, or unexpected, or called deviant, which is, excuse me, because they're infrequent, statistically infrequent in the sequence. Lost my voice there for a second. Okay. So auditory oddball is an example of something that's been studied a lot with brain network models. This is work from folks from originally UCL, Carl Friston and co, Marta Garrido, using dynamic causal modeling, which is a flavor of large-scale.
Using Janssen-Ritt models of the kind I described, and specifying a modest brain, large-scale brain network that describes well that models well the typical waveform that you see in this type of auditory attention paradigm. And these are interesting, and these are used in the context of psychiatry, neuropsychiatry, because you see differentiations in these evoked response patterns in patients with mental health problems like schizophrenia. Now, the main one I want to focus on here in the clinical applications is epilepsy. And I'm going to play the little video here to give you an idea. So epilepsy, as people probably know, is excessive uncontrolled neural discharges. So the time series here at the top is showing the depth electrode recordings. So these little dots on the brain here are implanted electrodes. The kind of lines of little dots. The little dots are channels across essentially a large pin that's inserted through the skull and recording activity from various points. But what you can see here in the time series is the points where we see these very large high activity periods the brown, and then it goes to blue, and then red and green. Those are seizures, and they spread. So just the fact we can see in the time series set on the bottom right. I mean, there are hundreds of channels here, but we're seeing a spreading from one part of the brain to other parts of the brain. And this is a typical feature of epileptic seizures: is they have a focus, an initiation zone, and then they spread. And the spreading is one of the is in a way the most dangerous thing, or the worst part of the seizure symptomatology, because it is what leads to things like grand mal seizures and loss of consciousness, loss of motor control, and so on. So one of the things that scientists want to do when they're so there are formulations of the dynamics of epileptic seizures that have been expressed in terms of neural mass models of the kind that we saw earlier, but these are neural mass models that generate time series that are that describe well the kind of time series dynamics, like in the bottom left-hand side here, of epileptic seizures. And one of the things that the folks, my colleagues in France, who've been working with this type of model in the clinical application to epilepsy, is they're using these connectome-based models of epilepsy, of epileptiform activity, to study and try to assess what the optimal place for a resection is. A resection is the surgical removal of brain tissue, which is the therapeutic option available when patients are not responsive to medication and have very serious seizures. So the surgeons need to remove some part of the brain. And what they typically want to do is they want to remove the epileptogenic zone, which is described here as EZ. But one of the things typically, well, sometimes you can't access the epileptogenic zone, or you don't know exactly. It might be in what's called eloquent tissue, might be some bit of tissue that you really don't want to remove. So you can then kind of default to saying, okay, well, if I can't remove the primary epileptogenic zone, maybe I can prevent the seizure spreading, the grand mal seizure, by targeting instead the propagation zone, which is described as PZ here. So this is one of the applications of brain network models in the epilepsy context, is trying to identify with a brain network model of epilepsy and epilepsy seizure spreading, where are the propagation zones, where are the epileptogenic zones, and where could how could this information be used to inform decision making in the context of surgical resections. And this is going ahead with clinical trials now in France, so it's really going quite far as an assistive technology, brain modeling based assistive technology. It's very impressive work from Victor Yersa and co. Other applications here, I'm just wrapping up because we're coming up to time now. So one of the other applications is in the area of stroke, and the general approach that you take here is we take our connectome, our brain network on the right-hand side, and what happens with stroke is you have bits of brain that die. Unfortunately, they become anoxic, and the gray matter and the white matter around the lesion, which you can see on the bottom middle here, the big black hole on the left-hand side, you have bits of brain that are just wiped out. So the way we represent that in brain network models is to take the graph, the connectome, and remove those bits of brain, either the gray matter, the nodes, or the white matter, the edges, and remove them from the network. And that's represented here on the right-hand side with these white lines that are kind of zeroing out the damaged regions. So we can study stroke from this network lesioning point of view with neural mass modeling. And final thing is brain stimulation, and this is something that I work on. So this is kind of close to home, and we'll see a bit of this in the practical tutorial tomorrow as well. So we can take our brain network model and we can take, say, additional information about the location that you apply brain stimulation. So the figure on the left-hand side here is just a schematic of what's called transcranial magnetic stimulation, which is a type of non-invasive brain stimulation where you basically put a large coil, a large magnet on the head, and you create a magnetic field that penetrates the skull and induces electrical activity in the brain, basically underneath the part of the skull where you applied the coil. And that's what this hot spot is here on the left-hand side and on this figure in the middle here. So we can say, and TMS, I should say, TMS is used clinically in a number of different areas, but principally in treatment of depression. So for patients who have major depressive disorder but are not responsive to antidepressant medication, TMS is a reasonably effective alternative therapy, and it's used quite a lot in Canada and in all over the world, in Toronto and in the institute where I work. But we don't understand so much about the physiology of it and the way in which applying a magnetic stimulation relates to and interacts with the brain at this physiological and network level. So what we can do, and one of the big focuses of the work that I do, is to take these connectome-based neural mass models, which we have this schematic here in the middle, this network. You've seen different variants of this, but we can say, okay, well, we know given where we're placing the coil and where we're injecting this stimulation, which is typically in called the dorsolateral prefrontal cortex and near the front of the brain. When we're injecting stimulation into there, where is it going? Where is a ping in the front of the brain propagating to? Because this is a highly dense and richly interconnected network, and this is something that we really want to know to improve our basic science understanding of brain stimulation, and ultimately to improve how we're developing and fine-tuning the use of this technology in the clinic. So we can use our brain simulations to simulate the injection of the current and then the propagation of the current throughout the rest of the brain. Try and understand better what's going on. And we will be looking at a modest example of brain stimulation simulations tomorrow in our practical session, which is going to be a lot of fun. Okay, so that's it for the lecture session today. Again, I will check over on questions, so feel free to throw some more questions at me, and we can have some discussion if people like. But just to wrap up, so we're looking at here large-scale whole brain network modeling. We're using a reduced level of description at the individual unit level, so we can think about neural populations rather than individual cells. And then by doing that reduction at the unit level, that gives us basically the computational real estate, if you like, to scale up in terms of the coverage. We can't do whole brain simulations at the level of individual cells. It's not computationally possible at this point in time. We can do whole brain simulations at the level of neural populations, and that is how we do it. So we obtain the network structure from non-invasive brain imaging using anatomical connectivity. We construct these anatomical connectomes, as we call them, and the dynamics plus the connectivity is the combination that we use to simulate brain activity. And these techniques have been, it's still early stages, the basic framework has been around for a long time, but the applications in the clinical domains are coming through over the last 5 to 10 years, really. So it's a really exciting area. There's lots of new directions, and there's people trying things out, and there's lots to explore. So it's a really exciting interface to be looking for how we can translate and use this type of modeling framework as a tool in the clinic and in clinical and cognitive neuroscience applications as well. So great, and more, sorry for the typo there, more in the practical session tomorrow afternoon. Wonderful. Okay, so I'm going to take a look at the chat and see how we're doing for questions. Okay, so I have one question on basic material. I can address that. I have another comment. Very cool. Thank you very much. I totally agree. This is very cool. This is why I do this. Great. So no other questions. So reading material. I think the best way of me giving you some good references is through the GitHub repo, which will be ready for you to peruse in a few hours. I have some final I's to dot and T's to cross. So I'll make sure that there's some references, but also some links to the references there. There are definitely some references in the Jupyter notebooks as well. And I'll even put some PDFs in the GitHub repo for you. I think I can get away with that. So yeah, reading material, basically check back. I'll circle back on this and give some comments on it tomorrow, but there'll be some for you in the GitHub repo. Which, to remind you, I guess, let's just give you the GitHub repo address for you to check. Although, as I said, it's going to be you're going to want to look in a few hours rather than right now. But in case you didn't catch it, there we go. John Griffiths, SciNet SS 2020 Brain Network Modeling. I'm seeing a question saying you're not seeing questions. Oh, yeah. I mean, I'm not seeing any more questions either. We had a question, a few questions, but not so many questions. So I can post questions privately. Am I getting, okay, have I had some private questions? I don't think I've seen any private questions. I'm sorry if I've missed some. But yeah, so to wrap up, I mean, any more final questions on the chat? Otherwise, we can call it a day for this session, and we can, you know, you guys can take a break, enjoy the rest of your day, and we'll see you again tomorrow afternoon at the same time. I'm flipping back to my concluding page because it's nice, but also because, oh, I've gone too far. Also because it has the URL for my lab, which people may be interested to have a little look at. So you can go to www.grifflab.com and you can see a little bit more about the work that I do, and that you can function as some background reading as well in the meantime. And you can find me on Twitter at Neurodidact. And the slides, I can make the slides available, and they can go on the course website as well. Okay, excellent. Well, thanks a lot, everyone. I hope this has been fun and informative, and I'm looking forward to getting cracking on the practical session tomorrow, same time.